Hi, I'm Ross Laws from Electron Microscopy Research Services at Newcastle University. I'm going to be doing some introductory videos to various image analysis software packages we use in our department, starting with Microscopy Image Browser. This is quite a nice tool developed by a team in Helsinki, and I'll start by showing you the website because that contains some useful information, and then show you how to install the software. So if you navigate to Google and type in MIB Helsinki, then it'll be the first result that comes up. On the main overview page, there is a section with the original publication. So the developers would ask that you cite the original publication in any papers where Microscopy Image Browser was used. So let's move on to the downloads. There are several versions of Microscopy Image Browser. You have the MATLAB version, which I will be using. This is a version that requires the full version of MATLAB to be installed for you to use it, and then you run it through MATLAB.
It's also worth noting that it requires the image processing toolbox, and as a recommended option there's also the parallel computing toolbox. Soon in the future there's going to be deep learning tools in MIB, so there'll be extra additional toolboxes needed for that as well.
But there's also a standalone version where you don't have to have MATLAB as well. And there's a standalone version also for the Mac, so there's a Windows and Mac standalone version. So I'm going to download that. I've already done this, so if you download it you'll get the MATLAB zip folder. What you can do then is just unzip that and place it in one of your files on your computer. I've just put it in my secondary program files here. What we can do then is go to MATLAB and the Home tab, then Set Path, and if you then navigate to where the path is for the folder by clicking this add with subfolders. This will add not only the base folder but then every subfolder within it as well.
It's worth noting you can just click add folder rather than add with subfolders if you want to keep the MATLAB path cleaner.
Right, so that's my folder. So I would select that and then it will add it to this. And then you click save. I'm not going to click save this time. All I need to do is click close because I've already done this. Then, once you've done that, you can simply enter MIB into the MATLAB command window. And the program will begin opening. You can't see the splash screen here because it's opening on the opposite screen. What you'll see when it opens up is the main UI and the tip of the day box. These can be quite useful when you're just starting off as they contain some nice little hints and tips. The first thing I'm going to cover in MIB is the user interface. So at the top here you can see we have the menu. There's a lot of options in here and I'll cover these as we go, as they're hard to explain without context. However, below that you have options to paste a dataset from the clipboard. You have the magnification tools here. You have undo and redo, which are useful. Then measure options. You can take a measurement and it will display it here and in the MATLAB command window as well. Navigate to that. Some of these last options require context to explain, and I can show you them as we go. Below the toolbar you have the section for picking the path where you wish to load your images from. Then below that once you've picked a directory, you have the directory contents section. This is where the contents of the folder will display. So in this folder, you will see a list of the folders and files in the current directory you chose. Below that is the segmentation panel. So here is where the model information is displayed. The materials in your model, so each material may correspond to a single object you've segmented or multiple objects. I'll explain more about this as we go. Below that is the tool section. You can select various segmentation tools here and then customize the tool below to apply whichever of these options you want. Next to that is the selection panel. Here you have all of the functions to manipulate your selection in different ways. On the right of that, view settings. Here are all the settings that allow you to change how you view your dataset. So you can show the model, show the mask, hide the image, change the transparency of each of these layers as well, and you can manipulate the contrast and display values of the base raw data. And on the right of that, finally, we have image filters. Okay, to load up a dataset, assuming this is your first time using MIB, you can click on this button here with the 3 dots on it in the top left-hand corner. This will bring up the Choose Directory window. What we can do then is pick the folder we want. Now, your images won't display in this directory browser. Don't panic, they are still there. This is purely just for picking the directory. When you have picked the directory, in the directory contents frame below, it will display the files and folders within that directory. If you can't see anything, once again, don't panic. It may be simply because you're looking at the raw data. The raw data from the microscope may be in one of the bio formats. Now, checking this checkbox here will display bio formats files. I'll include a link to the bioformats information page so you can check out which file formats are available in the bioformats library. It's also worth adding here that MIB has 2 reading modes. The default one opens all of the standard image formats plus a few of the ones that the developers regularly use, and the bio mode, which is, as I've explained, all of the bioformats library. Here we have a stack of images in the Gatan DM3 format from the Gatan 3 view, which we have. So to load them up, I'm going to select the first one, last one with Shift, and then right click, Combine Selected Datasets. Now, our images have loaded, and this is what you would do for any 2D stack or 3D stack of 2D slices like this. What you don't do to scroll through your images, I've seen this mistake made before, is click on the files here and double click to open them up. To scroll through your images, you will use this scroll bar that appears. So, just so we can see what we're looking at, I'm going to show you the display options. So, to change the contrast, you can do it automatically by just clicking minimum and maximum. That will get you an image you can see, not necessarily the best contrast. If you want the best contrast, you can adjust it manually and stretch the intensity peak across the histogram. Then we have very nice contrast on our image. You can also right click on the bottom and top end of the histogram to set the minimum and maximum that way,
which could be a little bit easier than trying to use the scroll bars to do it. Now if we scroll through, we can see what's happening with our image. If you want to load up a single 3D dataset, I have one in this folder here, so you can navigate to a folder within this directory I chose. Just double click. Now it's not a bioformat file, so I've got to uncheck that box. Then I can open up this dataset here. Now you can still right click and combine selected datasets, or you can double click and it'll open the image. Now this is just a post-processed version of the dataset we were just looking at. Once you've loaded the data, you can scroll through to check if there's any major anomalies that you might want to correct and remove later with post-processing and cropping. So if you're looking at 3D electron microscopy data, like SPF SEM data, you sometimes get darkening as you go through the dataset. That can be corrected with contrast normalization. We can't see it here because this is not a big dataset, this is quite a small one. However, at the start of the dataset, there is an anomaly here, maybe a cutting artifact or something from when we first started the data. The first step I'll try here is a contrast normalization. For that, you want to go into Image, then Contrast, Normalize Layers, then Z-Stack. So I would recommend doing this first on EM data, possibly on other data types as well, because what happens when you do the alignment step later on is it adds border padding around the outside of the image, so it can move the image frame around inside of that. What that border padding then can do is affect the way the contrast normalization algorithms work. So I'm going to stick with the default settings for this for now. For more detailed tutorials on how to use individual tools and features like this, I will make some other videos. So I'm going to run that. So you won't be able to see much of a difference here, because we don't really have a particularly large change in contrast throughout the dataset. So that can be quite useful, as I said, for EM data like ours. Can also be useful on confocal data if you're doing big stacks of immunostain cleared tissue and you want to they sometimes have a decrease in signal as you progress into the tissue, and also a decrease in stain intensity because of stain penetration. I would imagine this feature could be useful for that as well. Once the contrast normalization is complete, you can then lock in the display values that you want by going to Display again. Now, if you do any adjustments of the display like I did earlier, you will want to make sure you do it on a slice that doesn't have artifact on it, because that will probably be different from the rest of the slices. It might not be the case after it's been normalized, but it's best to do it maybe halfway through the dataset on a good slice like this. So I adjusted the peak earlier, so the histogram fit nice. The peak fit nicely into the histogram like this. So what you can do then is actually apply that so that this becomes the actual intensity graph for the image. So we're essentially only including this major peak. So I'm going to click all slices, and it's going to stretch that peak across the full
range of intensities. So 65,000 here to 0 here. And I would do this before, once again, do this before you do the alignment step. So next I'm going to show you the basics of the alignment for post-processing. So if we go to alignment tool, and we're going to leave everything on the default settings because we don't have any major misalignments or we haven't moved the image frame halfway through the run. It means we can just use the drift correction. So this is going to look for what it perceives to be, or what it determines to be, actual drift in the image, not sample movement because of the structure of the sample. So, in this case, for the background color I'm going to use black because it'll help better illustrate what's actually happening here. And I'm going to run that. This window, I usually don't use the running average, I just say yes. So what will come up in the end is a graph of the detected drifts. You can keep this and save it if you want to be able to include it with any data set. Say you've post-processed something which you end up actually putting into a publication. You might want to upload the graph with it, potentially, to show how the drifts were corrected and which drifts were detected. I'm just going to close this, since this is just an example. So what you can see is we now have this black area around the outside of the image. Now, really, this is all actually classified as the image now. So if you save it, this black area will be included. So what that means is your image has actually got bigger. It hasn't changed it hasn't resampled the pixels or anything like that. It's just made the image frame bigger to move this around in. But what you'll notice is, if you have a dataset that's particularly misaligned, this one isn't. It's only moved slightly. You'll be able to see that everything kind of looks aligned after that. So a few notes on the alignment step. If you have a bad section, now this is something that's only really common with 3D EM, particular SPF SEM data. I don't think it happens with FibSEM data, but what you can have with SPF SEM data is a section that's trimmed off with a microtome in the microscope will land on the block face and then distort and cover the region of interest for one or more images. If that happens, when the alignment algorithm reaches that point, it then can determine sometimes that the misalignment is erroneously extremely large. So say it might think that the next image is somewhere up here. In that case, it would make all of this region, which I can show you now, all of that area there would then be classified as the image. As you can imagine, that increases the memory size of the image vastly. So, if this occurs, it will fill the RAM and immediately cause a out-of-memory error in MATLAB. So, if that happens during the alignment, that's what is worth looking out for. What you'll see as well is the graph that we had before will not look like a jagged line, where with, like, say, a kind of random noise movement. It will look like a step, so it will just be a line and then a massive step, then another line. On the bottom axis, it shows you which frame it was up to, or it has the scale of which frame it was up to. So in the case of these images here, it could have happened maybe, say, halfway through, so it would have frame 50. You can use the graph to kind of figure out where the bad slice is if you didn't realize there was one in your data set.
The simplest way to remove one of the bad slices when you've found it is to go to Dataset, then Slice, and Delete Slices. What you can then do is either it will automatically take the slice that you're on, which is the easiest way, or you can manually enter the number of a slice in here. So I would just click OK after that.
So after you've completed the first 3 steps and you have adjusted the contrast, normalized the contrast, then adjusted the contrast after that, then done the alignment, you can then change the mode, image mode for your dataset to say 8-bit rather than 16-bit if you want to. This will reduce the size in the memory and when you store the data as well. So it's useful for speeding up operations you run on the dataset, and also for just decreasing the size that it's taken up on your hard drive. So I'm going to do that now. And what we'll see if we go into display, the graph now has been switched to the range for an 8-bit image, which is 0 to 255.
Switching the mode will readjust the histogram like I explained earlier when you click all slices. So you could potentially skip that earlier step out and just switch the mode instead.
So the next thing you could potentially do is crop the dataset. So if you go to Dataset, Crop Dataset, then there's multiple ways you can crop the dataset. We have interactive, manual, and from ROI. I'm just going to cover the interactive and manual methods for now. So for interactive, if you select it, there's a little description here that tells you how it works. I'll run through it anyway. So we can press the crop button to start, then click and drag to draw a rectangle of the area you want to crop. What's good about this is you can then scroll through the dataset and make sure that your image stays in frame if you want to as well. So what you can do is just exclude areas you're not interested in. Say if you've got one particular cell you want to segment at a time. This can make it a bit easier to handle the data rather than loading up an entire dataset. And then to crop that, you just double click inside of the box you drew. What we also have is this area at the beginning where we have a bad slice with some distortion on it. So I am just going to use the crop tool again, this time manually. So I am going to set the Z value here at 2 to 23. So that's slice 2 to slice 23. And then just click crop. So if we look at this now, we don't have that bad slice at the beginning. Finally, what you want to do is save your dataset after you've post-processed it. You don't want to have to run through the same thing over and over again every time you want to do any analysis or segmentation of this data. So what you can do then is go to File, Save Image As, and then you have a variety of options. I've already saved this, but I can save it again to demonstrate. I'm just going to give it a simple name, 1. You can use this format here, Amira Mesh Binary. This is compatible with Amira, the segmentation and 3D display program, so it can be quite nice if you want to just immediately load stuff into there as well. You can save it as JPEGs. I've never done this, and I never normally would because it's a lossy format. Hierarchical data format, H5. This is useful if you want to load data into iElastic. So you can do your post-processing in MIB and then load it into iElastic if you want. You've also got the MRC format, which is quite nice and universal as well, and it can be used in iMod. There's some lesser known formats down as you go further down the list. You can also do PNGs as well, and TIFFs, which are a lot more common for scientific images. There's two types of TIFF format here. I tend to just go with the uncompressed. If you do a TIFF, which I'll do first, then you get a few extra options when you go to save. So it's going to ask you this: what are the dataset parameters? In this case, we opened up the raw data, and our raw data from the 3 view has the voxel sizes already included, so they're not just in a text file or something, say, included with the image. So if you load up the raw data in this case, it will carry this over till you get to the point where you're saving your data. When the bioformats library is used, it should acquire the pixel sizes automatically from the metadata. It's also got the voxel size, which is 0.1, because we're dealing with slice thickness in the 3 view and in serial block face scanning electron microscopy. Then it's the slice thickness that makes the Z value. So it's always going to be bigger than the X and Y. Now, these are known as the voxel sizes. So a voxel is just a 3D pixel for anybody who's fairly new to image analysis. So that term will get thrown around quite a lot. So this means your voxel is 0.013 by 0.013 by 0.1 microns in size. So it's more of a cuboid in this data, but it would normally be a cube for other data types such as CT, MRI. Anything that takes a slice, even optical slices, can sometimes have a different sized Z. So just click OK. Unless you want to change any of the values. File name, I'm going to leave that as it is. Then do 3D stack. You can do 2D sequences if you want. However, this could lose the Z size if you do it as 2D. What you have to do instead, if you save your data as a 2D sequence of files, is load up the entire sequence. You can't have any more or less images. What MIB does is it encodes the physical Z height of the bounding box into the image, then divides that physical height by the number of slices to get you the individual slice thickness. TIFF files in MIB are limited to 2.6 gigabytes in size, so if your file ends up being bigger than that when you're saving it, it will end up causing an error. So we're just saving that as a single 3D TIFF. If we go into our process data folder here, uncheck bioformats, and we can see there it is. So that's one way you can save it. If you save it as any of the formats like the AMIRA format, then there's other options. For example, you can save as an AMIRA mesh binary file sequence. You can also save PNG and JPEG formats as a file sequence as well. I won't go into too much detail about the other formats for now. TIFF is a good basic one to start off with. If you want to use some of the other formats, I'll assume you have some knowledge of image analysis already. If you do want to save your images as a sequence of 2D slices, then all you have to do is go to File, Save As, and then what we want to do here is we select TIFF format uncompressed, save. Just going to use a new folder here for this. leave the voxel sizes as they are. Then switch this to 2D sequence and click OK. Once that's done, you're finished. So you've now saved your data and you're ready to move on to the next step, which is segmentation. So that concludes this first video. The next video will be on segmentation and the segmentation tools you can use in MIB.